So, today, we're going to dive into a pretty amazing piece of scientific detective work. It's a story about a really dangerous virus and a totally new way to fight it. And the idea is, what if you don't attack the virus at all, but instead you go after its secret partner in crime?
All right, let's set the scene. We're talking about the Chandipura virus, or CHPV for short. This thing is scary. It causes a severe swelling of the brain in children, and it moves with terrifying speed. We're talking high fever, coma, and often death, all inside of just 2 days.
And here's the big problem, right? For this virus, there's no vaccine to prevent it and no drugs to treat it once you're sick. So when you're up against an enemy that's this fast and this deadly, where do you even start?
Well, you start by thinking outside the box. Instead of chasing the virus itself, what if you could figure out the secret helper it needs to survive? And that leads to a whole new kind of strategy. It's called a host-targeted antiviral strategy. Okay, think of viruses like this. They're hijackers. They can't do anything on their own. They have to break into our cells and use our machinery, our own proteins, to make copies of themselves. So this strategy says, forget the hijacker for a second. Let's just disable their getaway car. And here's why that's so incredibly smart. When you target a virus directly, you're aiming at a moving target. Viruses, especially RNA viruses like this one, they mutate like crazy. That's how they get resistant to drugs. But our own proteins? They're stable. They don't change. They're a stationary target, which makes them a way better weak point to go after.
Okay, so we have the strategy. Now the real investigation begins. The mission: find the exact accomplice inside our own cells that this virus is using to do its dirty work.
And you know what? It didn't take long for all the clues to start pointing to one prime suspect, a human protein called Cyclophilin A, or CypA for short.
So why CypA? Well, the evidence was just piling up. First, its day job in our cells is to help other proteins fold into their correct shape, something the virus absolutely needs for its own parts. Second, scientists already knew that CypA helps out other similar viruses. And third, and this is the real kicker, there was already a drug on the market, Cyclosporin A, that was known to block CypA. So the investigators had their suspect, and they already had the handcuffs. Now it was time to prove it. The scientists set up this brilliant two-part investigation to really build their case, starting with a digital prediction and then taking it to a real-world biological test.
The plan was simple, but really elegant. First, they were going to use a computer to see if the virus protein and our host protein were a good match. Then, they take it to the lab to see if blocking that protein would actually stop the virus dead in its tracks.
So, first up, the computer. Using some pretty sophisticated modeling, they digitally smashed the virus's main structural protein right up against our human CypA A protein. And the computer said, yep, that's a perfect, stable fit. They could bind together really tightly. It was kind of like a digital smoking gun.
But, you know, a computer model is just a model. It's all ones and zeros. The huge question was, would this perfect match on a screen actually mean anything in the messy, chaotic world of a living cell during a real viral infection?
The answer was a huge, resounding yes, and this graph tells the whole story. See that blue line at the top? That's the virus just replicating like crazy, totally out of control. But then, look what happens when they add the drug that blocks our little helper protein. The red and green lines, that's the virus getting absolutely crippled. It's stopped cold.
Just how crippled are we talking? Well, the reduction was greater than 3 log. Now that's a technical term, but what it means is that the amount of virus dropped by over 1,000 times. 1,000. This isn't just slowing it down, it's a complete and total shutdown.
In other words, we're talking about a reduction of more than 99.9%. The scientists didn't just jam the virus's production line, they basically took away one of its most essential tools, and the whole factory ground to a halt.
So, after this incredible investigation, what's the verdict? And maybe more importantly, what does this all mean for the future?
Well, the verdict is in, and it's crystal clear. The prime suspect, Cyclophilin A, is guilty. It's a key accomplice, helping the virus's proteins get into shape so this deadly thing can keep replicating inside our cells.
So why is this such a big deal? Well, for one, we've found a chink in the armor of a virus that has 0 treatments. That's huge. Second, it's a massive win for this whole host targeting strategy. It proves the concept works. And finally, it gives researchers a concrete place to start.
They can now work on building new drugs that are specifically designed to break this connection, and they can even use existing drugs as a kind of blueprint.
And that leaves us with this one last really exciting thought. As we face down new and emerging viruses in the future, could this be the key? Instead of always chasing the mutating virus, could we just turn off the switches in our own cells that they need to survive? You know, this research suggests that might just be the future of fighting viruses.